Hello there and welcome to this Biology Mind A-Level tutorial on DNA replication. So just to recap, in our last lesson we learned about the structure of DNA and how to purify it. You now understand that complementary base pairing occurs through hydrogen bonds and that two anti-parallel chains of that DNA twist around each other to form a double helix. And nucleic acids from DNA can be purified via precipitation reactions. So we have three key learning objectives for today. Number one, why we need DNA replication. Number two, the actual process of it. And number three, the mechanism of DNA polymerase. So why do we need DNA replication? So it means that parents can pass their DNA to their offspring. Now, this passing of DNA and the genetic information stored in DNA is known as genetic continuity. Now, DNA replication occurs before the cell divides. So DNA replicates itself during the S phase of the cell cycle, so that each daughter cell has a copy of the DNA after cell division. So now let's have a look at the process of DNA replication. So I'll talk you through each of the stages, beginning with double helix unwinding. So the first step of DNA replication is this unwinding of the DNA, because remember it's structured as a double helix. Now, each of its strands is used as a template to form a new strand, and an enzyme called DNA helicase that you can see in this diagram here carries out the unwinding. So how does it do that? Well, it breaks the hydrogen bonds between complementary base pairs on the two strands of DNA, therefore allowing that double helix to now unwind because it's not held together by hydrogen bonds between its complementary base pairs. Now, the entire DNA does not unwind simultaneously. DNA replication occurs along an entire molecule of DNA, and the unwinding happens in one region of the molecule at a time, as you can see here, so only this part of the DNA is unwound, not this part. So this is done to ensure stability of the molecule. Now, the unwound region of the DNA is called a replication fork. Now, it's important to remember that DNA gets unwound in one direction only, meaning the replication fork moves continuously in a unilateral direction. Now, DNA replication has this property of being semi-conservative. So all this means is that the original strands of DNA act as a template for the synthesis of a new strands of DNA. So you can see that this is one strand of DNA and this is the second strand of the DNA. You can see one strand is here and the second strand is here, but they've both acted as a template for a new strand to be produced. Now, each new DNA molecule will be made up of one parent strand, so you can see that this is the parent strand because it's come from this original molecule of DNA, and likewise this is the parent strand because it's come from that same molecule of DNA. However, these are now the daughter strands because they are the new strands of DNA that have been produced from the parent strands, from the template strands. And how this happens is free-floating nucleotides in the nucleus are attracted to these parental strands of DNA, therefore ordering up in the exact format needed to make the daughter strand. Now, the newly attracted nucleotides are only hydrogen bonded with the parental strand, and condensation reactions between these nucleotides needed in order to synthesize the new daughter polynucleotide chain. And these condensation reactions are catalyzed by the enzyme DNA Polymerase, which reads the nucleotides and enables them to join. DNA Ligase is the enzyme that is responsible for the actual condensation reaction. So DNA Polymerase reads the nucleotides and enables them to join, and DNA Ligase joins them by condensation reactions. Now, DNA polymerase has this really important property of working in only one direction. Because remember, in DNA, one strand goes from the 3' to the 5' end, and the opposite strand goes from the 5' to the 3' end. So DNA polymerase can only bind to the 3' end of a parental strand and work in one direction. This means that they build the new daughter strand in the 5' to the 3' direction only, because if they're binding to the 3' end of the parental strand and they start working up the parental strand to the 5' end, that means the start of the daughter strand is going to be the 5' end to the 3' end. Does that make sense? Okay, now since DNA strands are anti-parallel but DNA polymerase can work only in one direction, replication has to occur in opposite directions on the two strands. And DNA is also being unwound in one direction only too. So the daughter strand, which will go in the 5' to the 3' direction towards the replication fork, can be made continuously. And this strand is called the leading strand. So this is the one that goes in the 5' to the 3' direction towards the replication fork. This can be made continuously. The other daughter strand runs in the opposite direction, so the 5' to the 3' end from the replication fork. So you can see here the 5' to the 3' end. This strand cannot be made continuously as DNA polymerase can only build the new strand in the opposite direction. So DNA polymerase will have to keep detaching and reattaching to this strand, resulting in the new strand being built in short segments. So this is the lagging strand, whereas this strand is built continuously, the leading strand. Now, when DNA polymerase binds to the parental DNA, it reads the nucleotide sequence and recruits complementary nucleotides. And while doing so, DNA polymerase carries out a proofreading activity. Now, this makes sure that only complementary nucleotides are pairing in order to prevent mutations from happening. So DNA polymerase has this function of being a proofreader as well. As the DNA polymerase recruits new nucleotides, remember I said that DNA ligase catalyzes the condensation reactions between the new nucleotides to create a polynucleotide chain. Now, just a note here is that some syllabuses do not mention DNA ligase while some do. And also, some textbooks say that DNA polymerase both has this property of proofreading but also catalyzing condensation, whereas technically speaking, DNA ligase catalyzes condensation. So if there's any confusion about this, I recommend reading mark schemes when doing past papers to see how marks are given to make sure that you get the right answer. Okay, this is our tutorial complete for today. Well done for following through, and I'll see you for the next one.